Hello friends, this class we are going to discuss one important aspect of biology, that is cell biology. From the school level we are acquainted with the term cell. At least 4 years back we might have been learnt what a cell is. Why in the beginning of the higher secondary level we are given so much of cell biology? Much is learnt about cell biology. At least one unit is given for cell biology. The thing is, cells are the fundamental units of life. All living forms, whatever you are observing in nature, let it be the plants or animals or the things you don't see, the bacteria, they are present in and around us. All are made of cells. So, cells are the fundamental units of life. All life forms are cellular. It means their life doesn't exist in any structure below the cells. So, cell is the basic. So, all life forms may be made of one cell or many cells or multicellular forms. Let me tell you why cell biology, that is the study of structure, function and behavior and relations of the cells, right? Because we can study all aspects of life, let it be metabolism, let it be reproduction, let it be function, let it be evolution, everything can be drawn to or focused on by studying cell biology. You might have learnt or listened the term cytology. Cytology, cytos means cell, study of cells. But now the term cytology is replaced by cell biology. Cell biology is a broad term that includes structure, function, behavior, evolution relationship and many more. Therefore, one unit at least covering 20 classes or so is given to cell biology, because we have to understand what cell is, because all biological explanations of life can be given if we understand cell structure, function, relation. When you are going to chemistry, much is studied or much time is given to study structure and function of atom. Similarly, biologists give more time on studying cell biology. So therefore, it is properly told, as atom is to a chemist, so cell is to a biologist. Similar the relationship. And famous cell biologist E.B. Wilson, he spoke, the key to every biological problem must finally be sought in the cell. If you have understood the cell properly, and in future you can answer all the aspects of life. Now, we have heard the name of Robert Hooke, who first coined the term cell. So he was a microscope maker, lens maker. At that time, the microscope, compound microscope, very primitive form, simple form of compound microscope he designed. With that, he visualized the slices of cork. Cork is the tissue of the bark, okay? It is the outer region. These cork cells of a particular plant he took, that is Quercus suber, and he made slices, thin slices as thin as possible, and observed through his own made compound microscope and found small cavities, and these are two drawings relating two samples, okay, and these small cavities reminded him the term cell. Cell means a room, a small size house, okay, and these things he published in 1665 in his book called Micrographia. You see, Micrographia, that means what he studied in microscope he published in the book named Micrographia. But the cell we are understanding today was not the same cell studied by Robert Hooke. Only he coined the term cell, but nothing about more about the cells could not be known by that time. As we see, day by day microscope improvement took place, staining techniques developed, new equipments came up. With this advancement in physical sciences, that led to development in biological sciences. So in that respect, almost after 175 years, that is in 1839, okay, cell theory was proposed. The two important persons, scientists, are Sladen, who is a botanist, and Swan, a zoologist. Let me tell you, Sladen, by profession he was a lawyer. Once he failed or performed poor in his profession, then he took interest in studying biology, botany, plant biology. So he studied the plant cells. He took different plants, sectioned them, studied them, and found plants consist of many cells. Plants are multicellular. Different types of cells are found in different tissues. Similarly, his idea of multicellularity of plants was extended by Swan, a professor in anatomy in zoology. So he extended his views and found similar results as Sladen found. He saw that animals also consist of cells. Then only Swan coined the term cell theory, and the two important concepts they gave, that life forms are made of cells. May it be unicellular like bacteria, or multicellular like we people or the plants. But as we see, another person, a pathologist, Rudolf Virchow, what he did, he saw that every cell arises from pre-existing cell. As plants come from plants, animals come from animals, so also cells come from pre-existing cell by means of a division. So this idea was included in the cell theory. The current theories relating cell theory or propositions relating cell theory are: the cell is the fundamental unit of life. All living organisms consist of one or more cells. We have learnt E. coli, Escherichia coli, it consists of single cell. Salmonella consists of single cell. Vibrio cholera consists of single cell. But we, the human beings, consist of nearly 10 to the power 13 number of cells, trillions of cells. So we are multicellular. Then life doesn't exist in units smaller than cells. That means subcellular structures do not exist independently. So a life to exist must have a cell. So all life forms are cellular. This must be remembered. Cells arise only from the pre-existing cells by means of division. The last tenet or proposition was given by Rudolf Virchow in 1858. Next. Then what a cell is? A cell is a membrane bound compartment. It has a boundary, it has a confinement that is provided by the plasma membrane, and within that it has protoplasm. In the protoplasm it must have the genetic material, and all cells in and around us have genetic material, that is DNA, right? So the plasma membrane that surrounds the cell maintains the integrity of a cell. A cell to exist, it must have its own boundary, that is plasma membrane. It encloses the protoplasm, the physical basis of life as proposed by Huxley. So where in this protoplasm all the reactions, all the biochemical reactions of life take place. Then a cell is an open system. A cell is an open system. What does it mean? A cell to grow, to divide, it requires raw materials, energy. From what source it get from? Now definitely it gets its energy and raw materials from the surrounding. Therefore cell is not an open system, not a closed system, it is an open system. Things are taken in and wastes are given out or products are given out. So exchange of energy and matter takes place between the cell and its environment through its plasma membrane. So now, and all the cells, all the life forms, they obey the laws of thermodynamics. Now let us see that how a cell works. Now the cell needs energy for this, and that energy is free energy, and that is free energy is fundamental to life. If you look at plants, plants take solar energy, convert this to chemical energy stored in the carbohydrate, right? When a cell stops utilizing free energy, it comes to death, or the process slows and ultimately it dies. And a living cell is a dynamic chemical system. Definitely it is not a static one, it is dynamic, it carries out work and operating far from chemical equilibrium. This thing must be understood by certain examples. Okay, operating far from chemical equilibrium. Actually at +2 level we have not gone to that level, but this thing I want to tell you. In the soil there is a citrus plant, lemon plant, and there is sugar cane plant. Both are growing in the same soil, same environment, but you see the differences of chemicals in both the plants. If you look at a leaf of lemon plant and the lemon itself, in the lemon the citric acid is stored in the vacuoles. You see, from soil to the plant and particular to the fruit, and particularly the cells where they store the citric acid, how the difference is. Another thing, calcium in the soil is very high, but in the plants it is very low. Potassium is very low in the soil, but in the plants it is very high. How the plants maintain this? That means they are not in equilibrium with the soil, with the environment. Rather, they are away from equilibrium, and that away from equilibrium things are maintained by the plants because of use of free energy. So all the reactions, it must be remembered, all the reactions of the living system are carried out by enzymes. They are the karak, semane karako. They carry out all the reactions, they mediate all the reactions. Without them no reactions can proceed fast. Okay, so all the enzymes, as we know, it is almost all are proteins. We will find some exceptions like RNA acting as proteins, RNA acting as enzymes in future classes. Okay, then come to this point. All the living organisms in this earth are grouped into two types. They are prokaryotes and eukaryotes. Prokaryotes and eukaryotes. Prokaryotes consist of prokaryotic cells. Eukaryotes are the organisms consisting of eukaryotic cells. So the term pro, eu and karyon, you can see this. Pro means primitive. We have seen protozoa and metazoa, the first formed animals, they are proto. The first formed cells, they are prokaryotes. Okay, bacteria are the prokaryotes. They don't have a definite membrane bound nucleus. Their genetic material floats in the general cytoplasm. But when we look at the plants, animals, fungi, protozoans, right, their genetic material, that is DNA, present in the chromosomes. The chromosomes are not present in the general cytoplasm, rather they are housed in a particular compartment called nucleus. Nucleus is a membrane bound structure, or it has an envelope within which the genetic material is suspended or remains. So therefore prokaryotes means before a nucleus. That means prokaryotes evolved first in this earth. If you study, prokaryotes arose when life came. Evolution of life or the first forms of life were the prokaryotes, right? So prokaryotes have a primitive cell, very simple cell, only a single compartment in which all activities goes on. Therefore its genetic material, which is a double stranded circular DNA, okay, it is floating in the general cytoplasm. So therefore when it is compared with higher versions of the cells called eukaryotic cells, eukaryotes have a definite nucleus, prokaryotes don't have that definite nucleus, that is membrane bound nucleus. That's why the term pro. So prokaryotes started, or they came to this earth almost 3.2 billion years ago at least. 320 कोटी वर्ष पूर्वे से माने पृथ्वी के आसी, and since then they are existing very much in this earth. But eukaryotes तार 200 कोटी वर्ष परे, that means आजि ठारू 120 कोटी वर्ष पूर्वरु eukaryotes मानकर evolution हेछि. So eukaryotes are young as compared to prokaryotes. So prokaryotes have faced all kinds of hot and cold, acid, alkali, salt, everything, UV and everything of this earth atmosphere or earth environmental conditions. But eukaryotes are, so to say, young as compared to prokaryotes, and prokaryotic diversity is very high as compared to eukaryotes. Now, so prokaryotic cells, in general, prokaryotic cells are small, simple in appearance, and they live mostly as independent individuals. One thing I can say, most prokaryotes exist as unicellular forms. We will see subsequently in certain photos, they are unicellular. Multicellularity is not found. If in certain case many prokaryotic cells, they are together, they are in the form of a colony, not they are interconnected. Okay, so they never form multicellular organisms, whereas they are either spherical in appearance or they are capsular form, rod form. They appear under light microscope. Okay, so often the prokaryotes, they are often protected by a tough cell wall, and below the cell wall there is the plasma membrane. The only kind of membrane, the only type of membrane in prokaryotic cell present below the cell wall is the plasma membrane, right? And the plasma membrane is the real boundary of the all the cells, so to say. Now within the cell wall or inner to the cell wall, it is the plasma membrane, and plasma membrane encloses the cytoplasm, which contains the genetic material, that is a double stranded circular DNA, and the RNA molecules, proteins, and the granular particles called ribosomes, which are involved in protein synthesis, and many small molecules needed for life. So under electron microscope, the cell interior appears under electron microscope. When it is seen under electron microscope, the bacterial cells appear granular in outer region. Let us have a look on the prokaryotic cells. One is a drawing, and another is, the second one is an electron micrograph of E. coli cell. The first one, if you read this, that is Vibrio cholerae. The bacterium, when it infects our intestinal tract, it causes cholera, and cholera claims almost nearly 1 lakh life earth in this world today. But this bacterium, as seen, it has a cell wall, the outermost thick layer. Inner to that is the plasma membrane, and you can see the granular structures. This is a drawing. These are the granular structures. So many number of ribosomes involved in protein synthesis, right? And in addition to that, there is a DNA. All prokaryotes, whether it is true bacteria or archaea, by otherwise told archaebacteria, whether it is archaebacteria or archaea or true bacteria, that is eubacteria, they are grouped under prokaryotes. They have a simple cell structure, and their genetic material is double stranded circular DNA. It has no ends, it is circular, and that DNA is compacted, coiled to form a compact structure called nucleoid. In eukaryotes, the term nucleus is there. Nucleus houses or encloses chromosomes, the genetic material DNA present in the chromosomes. So similarly, the genetic material, that is DNA, present in bacterial cells in the general cytoplasm. The region where the genetic material is found or concentrated, that region is called nucleoid, equivalence of nucleus. Nucleus contains genetic material, and this is also the genetic material. But the second diagram, it is a photograph, electron microscopic photograph. So that's why it is called electron micrograph. It gives high resolution. This bacteria E. coli, Escherichia coli, is sectioned, and when it is seen under electron microscope, and you can see the transparent regions, the cell's DNA is concentrated in the light staining region, and this is the nucleoid, and it is often called incipient nucleus or a very primitive nucleus, but it has the genetic material. Nucleus also has genetic material. Okay, and let me tell you, Escherichia coli, this bacterium lives symbiotically in our colon. That's why the specific epithet is called coli. And important thing is, whenever in the class any teacher goes for teaching on bacterium, he gives you a diagram like this, capsular form. And why not a circular form? Because this bacterium E. coli is a rod shaped bacterium, capsular bacterium, most thoroughly studied organism in this earth. Much data is available on this, and whenever we will go to DNA replication, translation, protein synthesis, many things, it is based on E. coli. Now In contrast to prokaryotic cells, eukaryotic cells, as the two cells under microscopy look different. One has a definite nucleus and the prokaryote doesn't have. And eukaryotes have nucleus in addition to this defining organelle. Eukaryotes जो सेल कहउछंदी बा ऑर्गेनिज्म कहउछंदी तांकू डिफाइन करूछि कि ना प्रेजेंस ऑफ न्यूक्लियस. सो न्यूक्लियस इज द ऑर्गेनेल दैट डिफाइन्स दिस ग्रुप. अंडर यूकैरियोट्स द ऑर्गेनिजम्स आर फंजाई, कबक आमे पढ़िछंती ओडिया रे, फंजाई, प्लांट्स, एनिमल्स, प्रोटोजोआ एंड सो. बट बैक्टीरिया एंड दैट अदर आर्किया they are under prokaryotes. Now eukaryotic cells are larger in comparison to prokaryotic cells, right? It is complex cell. As they came to this earth later, almost 200 crores years after, these are complex cells. They house their DNA in the nucleus. In addition to that, they have many internal membrane bound organelles, and many organelles we know they are mitochondria jahaku powerhouse of the cell कहती, plastids that is also the powerhouse of the plant cells, endoplasmic reticulum, Golgi complex, vacuole or lysosome, peroxisome. These are the membrane bound organelles within the eukaryotic cells. So it is appropriate to call the eukaryotic cell that is a compartment within compartments within a compartment. I have written it is a compartment of compartments. So these are small compartments and cell is big compartment, and all these compartments work coordinately, integratedly for the function of the cell, right? Then eukaryotic cells they are usually larger and 10 times in linear dimension, but geometrically if you look at by volume they are 1000 times larger than prokaryotic cells. So they have an elaborate cytoskeleton. We know our skeleton because when we are going to study the basic cell types, prokaryotes and eukaryotes, I told prokaryotes are simple, eukaryotes are complex. Eukaryotes have membrane bound organelles, they have large volume, and in addition they have cytoskeleton. We know our skeleton made of bones and cartilages, but cell skeleton is what? This concept of cell skeleton came after 1950s. Cellular skeleton or skeleton within the cell. मु गोटे कहबी जे आमे आमको देखुछंती, an estimate suggests that there are 10 to the power 13 number of cells in a human being. And when atlas is prepared of the entire human body, they come under 200 types. 200 टाइप रो सेल आम बॉडी रे अछंती. आमे तो जानी छंदी काहा को ब्रश बॉर्डर एपिथेलियम लाइनिंग द ट्रकिया, villi present in the intestine, rods and cones in the retina, brain cells, nerve cells. एमती जदि आम लिस्टिंग करती 200 टाइप आसवे तांकर आटलास बनइले देखियो सबु सेल देखिया पई मॉर्फोलॉजिकली दे आर डिफरेंट. बट मु गोटे क्वेश्चन पचारबी, ऑल आर बाउंडेड बाय ए प्लाज्मा मेम्ब्रेन, बट हाउ द सेल्स कैन बी डिफरेंट? डेफिनेटली द डिफरेंसेस इन मॉर्फोलॉजिकल स्ट्रक्चर इज एट्रिब्यूटेड टू बा ए जो डिफरेंस इन मॉर्फोलॉजी होउची कौन पाइना सेल मानक भितरे, प्लांट सेल आज वेल आज एनिमल सेल मानक भितरे, यूकैरियोटिक सेल्स मानक भितरे गोटे साइटोस्केलेटन रहुची. सेल स्केलेटन नॉट मेड ऑफ बोन्स एंड कार्टिलेजेस, रादर they are made of 2 to 3 kinds of protein filaments, proteinaceous rope like structures. They are chiefly of 2 types. They are actin filaments and microtubules. I will be telling later, but cytoskeleton are very much a signifying or significant feature of eukaryotic cells. In terms of size, prokaryotic cells range in 1 micrometer to 10 micrometer, and they are very small ones. But eukaryotic cells, they go from 10 to 100 micrometer, and some may cross more than 100 micrometer, right? And important is when compared, animal cells are smaller as compared to plant cells. About nuclear envelope, prokaryotes don't have, but they have the nuclear material, that is DNA. Both have, but it is not housed within the nucleus, that's why it is called nucleate, but true nucleus along with nuclear envelope is present. Third one, the genetic material is present as a single copy of double stranded circular DNA, and in some books it is written dsDNA, that means double stranded circular DNA, but in eukaryotes the chromosomes or the genetic material, that is DNA, is present in linear form. As we have 46 chromosomes, we have 46 DNAs. In each chromosome there is long DNA and there are two termini, two ends. It is not as circular in prokaryotes, it is linear. Okay. Another difference is, you might have heard the term chromatin, colored threads. Eukaryotic DNA, they are complexed with, associated with a class of proteins called histone. So histones when associated with the DNA, they give rise to chromatin. So therefore this is double stranded circular DNA, single copy, many DNAs representing many chromosomes, and this is naked DNA, but this is DNA associated with histone proteins. This is a very big difference between prokaryotes and eukaryotes. About organelles, can you say which organelle is common to both? It is ribosome. Ribosome is present in prokaryotes as well as eukaryotes, but they differ in size. We will study in detail while coming to the organelles, but prokaryotes have a small size and its size is 70s the unit, and it is 80s, right? And other organelles like mitochondria, chloroplast, rest of the membranous organelles are present here, but they are absent here. About cytoskeleton, as I discussed in eukaryotes, they are very unique to eukaryotes, they are absent in prokaryotes. Now about under eukaryotes, so many types of organisms are coming. If we group the organisms we have studied, I told prokaryotes under रे bacteria आसुछंती, जेते bacteria में पढ़िछंती, but eukaryotes under रे plants, animals, fungi, protozoa, right? This group is coming, 4 groups. They are protist, fungi, plantae, animalia, they are coming under eukaryotes. Now what this slide displays? This slide displays to acquaint with you about the major difference between plant and animal cells. The left one is an animal cell, the right one is a plant cell. The left one, as you see, it is of irregular shape, right? Not bounded by membrane. You can see the centriole, the labellings to left side or to right side, they are unique to the cells. Centriole is unique to animal cells. Centriole is the site, centriole and centrosome, they are unique to animals. They are the site of formation of microtubules. Such structures are not found in plant cells. Rather, what is the uniqueness of plant cells? Plants are autotroph. Definitely this must be coming to mind. Plants have plastids, green plastids, the chloroplast, right? Green plastids, the chloroplast. So therefore chloroplast are unique to the plants. Cell wall, which is outer to the plasma membrane, that is also unique. And moreover, another organelle called vacuole. Vacuole, sap storing vacuole, liquid storing vacuole, that is found in plant cells, but such kind of vacuole is not found in animal cells. So the major difference between plants and animals, plant cells have a definite shape and the shape is attributed to the presence of the cell wall around, which gives the definite shape. Then chloroplast and the group plastid gives color to the plants, make them photosynthetic, autotrophic. And vacuole, one thing you can see, one animal and one plant, if you compare, plants grow faster. What might be the reason? Plant cells are larger as compared to animal cells. The reason being, in animal cells, whatever stored is almost protoplasm, but major portion of plant cell is filled with vacuoles. So plant cells grow larger, quick growth is due to presence of or fulfilling or filling their vacuoles with more water. Okay. So this is the major difference. Subsequently, whatever difference in nutrition, locomotion and other sensory to the outer world, it differs from animal to plants because of the presence of cell wall, major. Now, as my criterion is to go in detail about the plant cells. Now, I have collected two photos. You see, the A1. This is, जदि आमे ओडिया रे बुझिबा, टगर गछ देखिछन्ती, तराट भी पढिछन्ती, तराट गोटे बड़ फुल टगर ठारू, तार लीफ रो एपिडर्मिस रे आम लैबोरेटरी व्हाट वी हैव डन, वी टुक दिस and observed it under compound microscope and we found the leaf epidermis, leaf रे जो skin रे एई भळा सेल माने मिलुछि. At least मु भाबुछि you people might have understood what these are. ओडिया रे जो पढिछन्ती आमे stomata are there. So the stomata, each stoma is guarded by two guard cells just like this, and they open during daytime and they close in evening and throughout night. And you can see there is 2 to 3 stomata which are open. Okay. Now these are the cells which have a definite boundary, and the important thing in this epidermis, all the cells are closely fitting. There is no gap in between. And the second one is the epidermis of fleshy leaf of onion. This is given to you people in the practical class in the first year to study the structure of cells. The easy material is onion. Onion रो जोटा में खाउछंती, जोटा को प्याज टा आनिला परे ताकू काटी भळा जोटा आसु स्केल लीफ ताकू छडी देउछन, आ जो पोर्शन खाउछंती, these are cells, storage leaves. Sorry, these are leaves, storage leaves. ताकू भांगिले से तुर जरी भळा लेयर बाहरिबो, किछि नाही. से जरी भळा लेयर को सिंपल माइक्रोस्कोप रे सॉरी स्लाइड ऊपर आमे ताकू वाटर देकी पकई दैछन, तापर टिके नाकू साफरान दियाछि स्टेन, बिकॉज सेटा एते ट्रांसपेरेंट रहे लाइट पास कर जाए, कंट्रास्ट आसे नाही. टू हैव कंट्रास्ट वी हैव added little bit of saffron in. So as a result which you can see the cells, because I have to go to plant cell. So now you can see this is the epidermis of a photosynthetic leaf. देखो यार होची जे एई लीफ रो काम होची फोटोसिंथेसिस. फोटोसिंथेसिस पाई गछर खाद्य होची कार्बन डाइऑक्साइड. लीफ रो स्किन टा जदि कंप्लीट हे जो कार्बन डाइऑक्साइड जाई परिबो नाही. फॉर दैट रीजन इट मस्ट हैव स्टोमाटा, पोर्स, एंड ए पोर गुडा दिन बेले खोलंती, कारण दिन बेले सूर्य किरण आसंती, खाद्य तैयारी हुए, सेथि पई कार्बन डाइऑक्साइड जेबा दरकार. यू सी हाउ द कोरिलेशन बिटवीन द structure of stomata guarded by two guard cells, they open during daytime to allow entry of carbon dioxide which is used for photosynthesis, जो आलोक संश्लेषण आम पढ़ा, right? And other cells do not allow any exchange, they are as a barrier, and this is from a storage leaf. There is no need of photosynthesis. से गुड़ा माटी तले रहे पियाज तेणु तार photosynthesis होबार नहीं. But thing is you see how आमे जदि देखिबा से सेल मानक भितरे जो सेल वॉल कॉमन वॉल केते पतला सेटा स्टोरेज तेणुकर सेठे शक्त हवा दरकार नहीं सेल वॉल मोटा हवा दरकार नहीं. I want to impress you people that the storage cells which are soft cells they have thin wall, because we will be coming to plant cell wall in few minutes later. Okay, so now we will be discussing the structure of a typical plant cell. Have you heard a typical plant cell? You might have studied photosynthetic cell, root cell, leaf cell, likewise. But what a typical cell? It is from type cell. Type to typical, because I gave you impression or I shown you two photos. One is of onion, the other is from a leaf. But there you found three types of cell. Guard cells which surround the stoma, other epidermal cells, they are of different outline. Third one is onion leaf cells, they are more long than broad, almost pointed at the ends. And if you look at or study in future plants from roots, leaves, stem, bark, you will find numerous type of cells, but all are surrounded by wall. Okay, so what cell we will be taking, whether we will study xylem cell, tracheids cells or basal cell, or photosynthetic cell or leaf cell. Therefore, taking all the aspects of cells, we have a generalized plant cell, type cell, ideal cell, which will reflect the cell structures of all cell types. So now this typical cell nears to a parenchyma cell, a photosynthetic cell. Okay, the features of this cell is very near to your parenchyma cell, photosynthetic cell, right? Now look at this. The first, if we are coming from outer to the interior of the cell, the outer structure is the cell wall. Okay, cell wall is a thick structure present outer to the plasma membrane. It is much thick. Plasma membrane is very thin. Plasma membrane is very thin. Therefore, so plasma membrane encloses all the protoplasm. Now the two terms can be described. One is cell, plant cell can be divisible into cell wall, and the contents entirely enclosed by plasma membrane is protoplast. Then protoplast contains a major organelle, the master organelle, that is nucleus. So the protoplasm can be divided into the cytoplasm and the nucleus. Then the cytoplasm has different organelles, as you see, the chloroplasts, mitochondria, endoplasmic reticulum of two different types, Golgi complex, cytoskeletal elements, very small bodies, micro bodies. So all these, when taken into account, these cytoplasmic organelles, they are floating in a ground matrix, that is called your cytosol, right? We will be describing all these in detail, but a typical plant cell houses or contains all these organelles. You may not find this chloroplast in root cells. They will be having different kinds of plastid, but we have to speak what is seen under electron microscope ultimately. That is ultra structure of a typical plant cell, fine structure of a type cell, generalized plant cell. Now this, you can look at this table, the plant cell, protoplast, cell wall, and whatever the shape of the cell may be, may it be hexagonal in outline, or spherical or more columnar. When cell wall is removed, the protoplast becomes a sphere, because that sphere fits according to the cell wall. So cell wall provides the shape. Then plasma membrane and protoplasm, as we discussed, protoplasm has cytoplasm, the nucleus, the vacuole, and the ergastic substances. Everything would be dealt detail, but if we go in this way, categorizing all the protoplasmic structures, then it would be easy for studying the cell structure in detail. Okay, the cytoplasmic organelles are endoplasmic reticulum. You must remember, reticulum, it is a network, as it is present within the protoplasm. That's why the name endoplasmic reticulum, right? And it constitutes almost 90% of the entire cell membrane. Then Golgi apparatus, which is involved in secretion. Lysosome, very often we call it the suicidal bag, but we will see its role, what it plays. But in plants, lysosome, the term is not used, rather vacuole is used. We will see. Mitochondria, the powerhouse of the cell, it generates ATP. Plastids, especially the green plastids, they convert this solar energy, radiant energy, photon energy to chemical energy of carbohydrate. And these microbodies, they have different roles. Ribosomes, the workbench of protein synthesis, and towards the end I told you the cytoskeleton. Already I have told what role they play in the cells, right? The plant cell, the same description. The plant cell is typically consists of a rigid cell wall. It encloses a protoplast, and protoplast encloses the ground substance, that is cytoplasm, and the major organelle in the protoplasm is your nucleus. Okay, so these things are the descriptions. Plant cells develop one more, often liquid filled cavities called vacuoles. In Odia we have read it. Odia taku gote term vacuole को कहुचे रसदानी से रस स्टोर हो खाली गोटे जिनस कहि आम आखु खाऊँ आखुर जो रसटा सुक्रोज डिजल्ड इन वाटर अल्टीमेटली कौटे रोचे ना आखुर से जो स्टेम तार सेल सेल भीतर वैक्यूअल भीतर रोचे लेंबू लेंबूर जो सिट्रिक एसिड व्हेयर इट इज स्टोर्ड ना इट इज इन द वैक्यूअल. So from these two I can tell you vacuoles stores liquid and dissolved substances in it. It has many roles. It taku in general कहती it is the dump yard of plant cells. कहीं ना animals मानकर regular basis रे excretion हुए, but plants don't go for this for excretion, but their wastes do they don't excrete it to outside. Where they store, what they do with? Now definitely they dump their waste into the vacuole. Vacuole is a major feature of plant cells. It is generally called central vacuole because it is a big vacuole, central vacuole, but small vacuoles having different roles are found in animal cells. We will be encountering or we will be discussing them. Now let us come to the important feature of plant cells, that is the cell wall. Cell wall is often called extracellular matrix because it is present outer to the plasma membrane. It is thick. Okay. So all plants, when we study the plant cells, they have wall present outer to the plasma membrane. When studied from different cells, the cell wall consists of different layers. Let it be like this: middle lamella, inner to that primary wall, inner to that wherever present it is secondary wall. Now what middle lamella does? Middle lamella generally works as a cementing substance. It is made of pectin, which is highly hydrophilic. It stores water. along with this pectin it contains magnesium, right? Now how this, and calcium, how this pectin and this divalent cation that acts as a glue, दीटा सेल को धरी रखुची, यार सेल वॉल भीतर जो थीन लेयर अठा बड़ा सब्सटेंस रहुची, दैट इज योर कॉल्ड मिडिल लेयर बा मिडिल लामेला. मिडिल लामेला इज अ ग्लू अठा. इट बाइंड्स टू two primary walls as we will be observing from the picture. Then inner to that is primary wall. Why it is so called primary wall? As it forms first primarily during cell division. So primary wall develops first, that's why it is called primary wall. Remember, primary wall, secondary wall. Do all cells have primary wall as well as secondary wall? No. All cells have primary wall, but all cells don't have secondary wall. I will be telling which cells develop secondary wall, but all cells have middle lamella inner to that primary wall. Okay, and a primary wall is thin, highly hydrophilic. It is made of cellulose, the most abundant biomolecule. It is fibrous, then hemicellulose, and pectin is there, and some amount of glycoproteins are there in the primary cell wall. Okay, look at this. This is a drawing and that is a photograph. Now this is a computer generated model. This is the middle lamella. When we join two bricks, a special motor जो सीमेंट दिया हुए अल्प दिया हुए, कहीं ना सेटा अठा. Glue is very thin. So this is pectin and magnesium, and inner to that in this green is your primary wall. Primary wall is thin, gets deposited first. Then secondary wall. Remember, जो पियाजर सेल देखई थिली सेटा होची प्राइमरी वॉल, बिकॉज दे पोजेस प्राइमरी वॉल. सेटे सेल वॉलटा जना पड़ू नथला. दिहिंकर सेल वॉल टोटल इज वेरी थिन. नाउ सेकेंडरी वॉल forms in those cells which have stopped growing, and strength is must. If you remember the fiber cells आम सजना छुई की देखा. सजना छुई जो पल्प रे जो सेल्स माने अच्छे दे हैव प्राइमरी वॉल, किंतु द फाइबर कोकोनट फाइबर से जो सेल्स माने अच्छे से माने होछन थिक वॉल्ड सेल्स. से सेल्स मानकरे प्राइमरी वॉल तलकू इनर टू द प्राइमरी वॉल बट आउटर टू द प्लाज्मा मेम्ब्रेन. दिस मच is the secondary wall. Secondary wall is thick. It has more of cellulose and lignin. Secondary wall is thicker and more important, it is strong. Where secondary wall gets deposited, where strength matters, where strength is necessary, elasticity is necessary, in those cells secondary wall gets deposited, but primary wall is everywhere. So now in plant cells, this is the description. There is rigid wall and the functions, these are the functions. They provide shape to the cell, and important thing is that they store much water in them. I told you vacuole stores much water, or vacuoles are almost filled with water and certain minerals and proteins like this, but the cells are prevented from bursting, or they do not burst because of this cell wall. So cell wall prevents osmotic lysis. And second wall is cell wall. When you deposit, think of the structure, cellulose fibers are arranged just like in a ply like manner. You look at my hand, these are the fibers, one layer, then another layer, and there you will be getting the pores. So wall is porous, therefore wall which is present outer to the plasma membrane is porous, permeable. This must be remembered. cell wall is porous and permeable through which water and molecules, small molecules can easily permeate, they can easily pass and go to the plasma membrane, but plasma membrane is selective in function. To the end, let me tell you, it is a structural barrier to the pathogens. आमे सबु गछ को देखिछंदी, गछर बकला से सेल से किछि न थाए, से सब डेड सेल्स, बाहर गुडा डेड सेल्स एंड तार भीतर जो प्रोटोप्लास्ट मरी जाई थाए, ताहले जेते बेले कोनसे गोटे ऑर्गेनिजम ताको कामुडी जीव ता भीतर लिविंग सब्सटेंस रस किछि नथिवो, सेथि पई केही ताको सहजरे खांती नाही. ता माने मु कहया पे चाहूची, व्हेयर एवर सेल वॉल इज ओनली प्रेजेंट and cells are dead, they are distesting, they don't give any taste, they don't give any nutrition to any pathogen, bacteria and fungi. That's why it is cell wall is used for protection. Thank you. This is the electron micrograph of the fiber cells. This fiber cell is a sclerenchyma cell. These are long cells and they give strength and elasticity to the plant. So this is from a taxus, that is from a gymnosperm, जो नग्नबीजी गछ पढ़ी तारी गोटे. You can see this is the common region. So here lies the middle lamella and the primary wall, right? एठी देखे, this is the middle lamella and this is the primary wall. ता तळगु देखो secondary wall, secondary wall, secondary wall, S1, S2, S3. So when secondary wall is deposited, first layer, then second layer, wherever it is needed, but ultimately when walls are formed, आमे जदि घरे बहुत सारी प्लास्टर लगे लगे जीवा ताहले कोन होबे, घरर वॉल्यूम टा धीरे धीरे कमी कमी आसीबो. से भलिया जते सेकेंडरी वॉल जते जमा हे आसे, डिपोजिटेड हे आसे, ultimately the functional volume or the lumen of the cell get diminished, and these are mechanical cells. So that's why having protoplast at the end is meaningless, but their purpose is to provide strength and elasticity. Okay, this is about the cell wall. Thank you.